Hi, everybody. Welcome back. This is the first mini lesson on Chapter 5, which is microbial metabolism. So this first mini lesson is going to be covering just an overview of metabolism. I'm calling it just presenting some general ideas, hopefully rehashing some prior knowledge that you might have learned in other classes. So we're just going to kind of get some groundwork, and then we're going to jump right into glycolysis and the rest of the processes of cellular respiration. We'll touch on some fermentation and photosynthesis to wrap up this chapter. This first picture I usually show in class, just kind of as a way to just remind everybody that what we're looking at is just the very kind of skeletal processes that are kind of the key events that take place in metabolism. So for example, this process right here in the middle, that's glycolysis. So we're going to be running through glycolysis, and just those 10 steps are part of this huge poster of all of these other different metabolic pathways, and every arrow that you see is probably catalyzed by an enzyme. You can see intermediate products going over here. You have some things coming in here in different ways. You have some of these things going into other directions. So just a good visual representation to say, while we might be seeing these things kind of isolated from other reactions, just know that, remember, inside of a cell, it's a big bag of cytoplasm and enzymes and metabolites and intermediate molecules and products and reactants and all that kind of stuff. And there's no rule to say this particular glucose molecule will stay in all 10 steps to produce these 2 pyruvates without interacting or interchanging with other things. So that's kind of the purpose of why I do this picture, not to be overwhelming, but just to let you guys see that what we're learning is just a kind of a sliver into the reality of what microbial metabolism, or just metabolism in general, is like. So when we take a look at metabolism, it is the term, if we want to define it, is the collection of all chemical reactions that take place inside of a cell. Okay, there's your textbook definition. And it is made up of 2 halves of chemical reactions. We have catabolic pathways, which are breaking things down, and anabolic pathways, which are building things up. So we are going to focus primarily on the catabolic pathway of cellular respiration, which is breaking down a molecule of glucose. We will see an anabolic pathway with photosynthesis, which will likely be new to most people since we do not cover photosynthesis in our Bi 211 here at RCC. So with this idea of metabolism, this is kind of another overview slide. In the notes, there's a list of 8 elementary statements that are illustrated kind of by this diagram. So I'm going to go through and number those on this diagram. So the first one says every cell acquires nutrients. So that's kind of this step 1. Whether you are a bacteria, we'll just stick with bacteria, but I mean, all of these are true for eukaryotic cells as well, but it's micro, so let's talk about bacteria. Whether you are a bacteria that consumes nutrients from your environment, like diffusing them into your cell, or whether you are a photosynthetic organism that generates your own glucose, this is your nutrients, right? You're getting them from your environment or you're making them yourselves. The next step is metabolism requires energy from light or catalyzed nutrients. So here's our catabolism, and here's the energy. Some of that energy is lost as heat. Some of that energy is stored into ATP. So this is going to be our step 2. We get energy from these catabolic pathways. They are exergonic processes, and energy is released from those. Statement 3 is energy is stored in ATP. Right? So as we catabolize nutrients like glucose, which we're going to do for cellular respiration, we break that down, we release the energy. The energy is stored for the most part in ATP. We always are going to lose a little bit of it as heat. That's just the nature of chemical reactions. Number 4, cells catabolize nutrients to form precursor metabolites. So not only are we breaking those nutrients down to harness and extract the energy, but we're changing their chemical form to be from like a large carbohydrate to maybe an individual carbon atom, or a large protein into individual amino acids, or a large fat into individual fatty acids. And in the process of extracting energy, we are also making these precursor molecules that can then be used in other processes in the cell. Number 5, these precursor metabolites, energy from ATP and enzymes, are used in anabolic pathways. Okay, so that's our energy that we just harnessed from our catabolic pathways are being used to drive our anabolic pathways with our precursor metabolites. Energy enzymes are going to make our larger molecules. So that's number 6. The ATP, the precursor molecules, our end result are these larger building blocks like macromolecules. So we say we eat a protein, we break it down into the individual amino acids, and then we build it back up into the protein that we want by using those amino acid building blocks. Number 7, cells grow by assembling macromolecules into cellular structures. So that's 7, kind of the branching there. And we can store those as well. So I'm going to put 7 kind of over here. So either you can use them to make more of what you want, like a new carbohydrate or a new protein channel, or you can store them, right? So bacteria have inclusions, those little piles of stuff inside of the cell. They can store those carbohydrates or lipids, which can then also be your source of nutrients, which starts the whole process over again. And then lastly, the whole point of this in bacteria is for cells to divide, to make a copy of their DNA, to split in half, and then you start, each new daughter cell can start the whole process again. So these are those 8 basic statements, elementary statements of metabolism. You take in nutrients, you break those down, you release energy, you store that energy, you use those precursor metabolites with energy to make larger building blocks, you build more of your cells, and you divide. Simple as that, right? That's the life of a bacteria. Okay. The next kind of key topic that we need to understand before we start applying this to metabolism is the idea of oxidation reduction reactions, also known as redox. Kind of an abbreviated form. So teachers can say redox instead of oxidation reduction reactions. Okay. So the idea for redox reactions is there are going to be 2 things. In this case, the orange circle and the green oval are the molecules of interest here. One of them is going to give off an electron. And if you are unfamiliar with the atomic structure, it's been a while since you've had your general or basic chemistry ideas, you can go back and check in chapter 2, I think in this textbook, it does a little bit of a review of chemistry, knowing that electrons, those valence shell electrons are involved in chemical bonding, and in ionic bonds, they can be given off and accepted to fulfill the octet, all those wonderful basic chemistry ideas. If you need to review that, feel free to go back to chapter 2 in your micro text, chapter 2 if you have the A&P text, and chapters 2 and 3 if you still have your by 211 biology text from RCC. Or send me an email or come to office hour, whatever. But in this idea of the redox reactions, one species of molecule is going to give away an electron, and then another species of molecule is going to accept that electron. That's all redox reactions are, is the transfer of electrons from one molecule or atom to another molecule or atom. So we just now label who's who. If you are the substance that's donating the electron, you're giving off the electron, you are being oxidized. Losing electrons is oxidation. The thing that's gaining the electrons is being reduced. So gaining electrons reduction. So I have my Leo the lion says Gur saying, so losing electrons is oxidation, gaining electrons is reduction. So hopefully that will help you remember what's happening here. So the orange guy, we'll just say A, with its electron, is donating that electron, and it's just A, usually plus, because it's lost an electric charge, and not always, but typically when you lose a negative charge, that basically means you've either gained a positive charge or you're less negative. Then we have green oval here that's gaining an electric charge, which means he might be now negatively charged, or if he was negatively charged before, it's going to be neutral now. So when we identify these molecules, you're going to be looking for clues of who's losing the electron, who's gaining the electron based on maybe charges. Another thing to remember is electrons themselves can't really go from one place to the next all by themselves, isolated. They're usually going to be hooked onto a hydrogen. So remember, a hydrogen atom in the nucleus has 1 proton, no neutrons, and 1 electron floating out around outside of it. When a substance, the electron donor, donates the electron, it's usually going to be housed with a hydrogen atom. So basically this is going to be like a hydrogen atom with a negative charge to it. It's got 2 electrons kind of hovering around that proton, and that's what's going to be moved to your electron acceptor. And so looking for clues of who is being oxidized and who's being reduced is you're looking for where the hydrogen with the electrons are coming from. So that's also another clue. We're going to be seeing redox reactions in every step of cellular respiration, as well as in photosynthesis. Glycolysis, acetyl-CoA step, Krebs, electron transport chain. We'll see it in fermentation, as well as photosynthesis. So it's a really important reaction. Again, if you are unsure or if it's a little bit not super solid in your understanding, make sure you get some assistance with helping you understand redox reactions, because that's really important to understand the rest of the process. Okay, and then lastly, our understanding of ATP. Hopefully, again, you should have some of this as prior knowledge. When we have adenosine triphosphate, that's this guy right here. It is made up of 3 parts: the nitrogenous base, the sugar, and 3 phosphate groups. And you might say, well, that looks a lot like a nucleotide. Well, it sure does, because that's basically what it is. It's an adenine nucleotide. An adenine triphosphate it's the adenine base. The sugar looks like it is an RNA, ribose sugar, with 3 phosphate groups hooked onto it. It is the energy in this bond here between the 2nd and the 3rd phosphate that's this high energy bond that when that is broken, it's exergonic, it releases energy, which helps to drive chemical reactions. So the way that we generate ATP is by the processes of catabolism, right? We're taking in nutrients, we're breaking those bonds, we are harnessing that energy that was stored, say, in the sugar, the protein, or the fat, and storing that by hooking that 3rd phosphate group onto an ADP to produce the ATP. So larger molecules getting broken down into smaller molecules is basically the definition of catabolism. And in doing so, we store that energy from the large molecules into ATP. On the flip side to that, if we want to build up larger molecules, say take individual amino acids and build a large protein, we have to go from smaller molecules to larger molecules. That is an endergonic reaction. You have to have an input of ATP. So all the ATP that you just made by breaking down nutrients, you can release that 3rd phosphate group, right? So now it's missing. And in that process, that releases energy to drive these anabolic reactions. Okay? So this is the cyclic nature of ATP. Your cells, bacterial cells, your cells, cat cells, plant cells, slime cells, they are all doing this cycle of making ATP and breaking ATP and making and breaking and making and breaking, because that's how metabolic pathways work in cells, both prokaryotic and eukaryotic cells. In this process going this way, when we are catabolizing and releasing the energy stored in those nutrients, we are making ATP. And our cells, not our cells, but cells in general, have 3 ways of doing that, these enzymatically driven ways. We have what's called substrate level phosphorylation, oxidative phosphorylation, and photophosphorylation. And the wording, the names might give you clues as to what's actually going on. Substrate level is like, enzymes and substrates. It's a direct connection of producing ATP just by adding that 3rd phosphate group onto the ADP right there, with usually just like substrate, like a intermediate. We'll see this in glycolysis. I'll point that out when we're doing each one of these. Oxidative phosphorylation is the electron transport chain and chemiosmosis at the end of cellular respiration with the redox reactions, oxidative, 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 driving the production of ATP via chemiosmosis. And then photophosphorylation, as the name implies, is a process that takes place during photosynthesis, which we'll see in the last lecture of this chapter 5 collection. All right, so that's our overview of kind of what is metabolism, some basic statements to lay the groundwork, your understanding of redox, and your understanding of how ATP works. So we will jump into cellular respiration with glycolysis is going to be our next video. Okay, see you next time.